to the organizers for inviting me. I really enjoy visiting China, and it's my first visit since COVID, so it's really nice to be back. And also really nice for me personally when I come to China to meet my former trainees, who now many of them are pursuing careers in academia and industry here. So very nice. All right, so I will talk about comparative biology. Unfortunately, Steve Oster chose not to speak about this subject because he would have given the best introduction to the topic, so I will try. So, although what Steve did say is that choosing species is important, so I will really now expand on this topic. So yeah, most of us use short-lived model organisms in our research, and we do this too, so I will not say, there is something wrong with it. They are convenient, but at the same time, if we only focus on short-lived organisms, we may really miss the mechanisms of longevity because, well, humans are one of the longest-lived mammals, so almost the longest. There is just one mammal that lives longer, and I will talk about this mammal today. So those short-lived organisms may not even have the genes or mechanisms that would extend human lifespan. That's why it is really important to also look at long-lived species. And if we only focus on mammals, there is tremendous diversity of lifespans. From very short-lived, well, we have the mouse here, and shrew lives even shorter, all the way to longer-lived animals. So here is naked mole rat, one of our models, that is a mouse-sized rodent that lives 10 times longer than mouse. We will study bats that can live into their 30s and 40s, and all the way to the bowhead whale that is the longest-lived mammal, and bowhead whale can live more than 200 years. So if we understand the mechanisms that are different between these species and these species, then we can apply those mechanisms to benefit human lifespan. So this is sort of the paradigm that we are using in our research, where we study long-lived species, we identify the mechanisms, and then we try to model it in mice to see if the mechanism can be exported to the mouse, and if it is successful, so then we can think how we can achieve the same result pharmacologically. And I will today show you several examples of long-lived species and what they can teach us. So here, I will start with naked mole rat. So now, 10 years ago, we discovered that high molecular weight hyaluronic acid is very important for naked mole rat cancer resistance. So naked mole rat, as I already mentioned, is quite remarkable. They live more than 40 years. They are resistant to all kinds of age-related conditions, from cancer to heart disease, arthritis. We keep them in group housing. They are not pathogen-free, but although their very long lifespan, of course, puts some limitation into what we can do. But at the same time, they're a wonderful discovery platform. So once we find something interesting, then we can test it in mice. So after we discovered high molecular weight hyaluronic acid, next question we asked was, can we engineer it in mice? So we made transgenic mice that express hyaluronan synthase gene from the naked mole rat, and we managed to slightly increase the level of high molecular weight hyaluronan in these mice, not to the same level as in the mole rat, but nevertheless, these mice showed reduced inflammation, which then resulted in increased lifespan and increased health span. There were several mechanisms that were related to immunoregulatory effect, enhanced gut barrier function, and antioxidant properties of high molecular weight hyaluronic acid. But overall, I would say this is sort of a proof of principle that we can take a mechanism from naked mole rat and then bring it into mice, and it actually works there as well. So although, well, it took us 10 years from one study to the next. But, that was a success story. And now the next step, of course, is how do we translated to humans. Of course, gene therapy is one strategy, but well, there are dangers associated, and pharmacological strategies, of course, preferred. So here we searched for inhibitors of the enzymes that degrade hyaluronan, and this way we can increase endogenous level of hyaluronan. We identified such molecules, so one of them, which is actually a natural product, when we administered it to mice and also injected them with tumor cells, it slowed down metastasis in the mouse. So we can already show that with these molecules we can at least have impact on cancer, and aging test will be the next step. So here we went all the way from naked mole right now to pharmacological strategies. So now I want to show you another example. So here we looked not at one species, but at multiple. And we were looking to identify a mechanism or a pathway that is consistently enhanced in longer-lived species. This paper was published in 2019, and what we found was that double-strand break repair was very strongly correlating with maximum lifespan in rodents, and not everything correlates. So we looked, for example, at base excision repair, nucleotide excision repair. We didn't find very strong correlation. There was a mild one, but modest. There were other factors. For example, amount of light exposure was more important for nucleotide excision repair. But double-strand break repair really correlated with lifespan. And then when we searched for the mechanism, we found that it correlated very strongly with the activity of sirtuin-6 in these species. And longer-lived species had more active SIRT6. We could even swap a few amino acids between mouse and beaver. So for those who don't know, beavers are very long-lived. They can live more than 20 years, or maybe even more than 50. So we were able to change activity of sirtuin-6 and improve DNA repair. So that was very promising. And then also combined with our finding of allele that was enriched in human centenarians, allele of SIRT6, that enhanced mono-ATP ribosylation activity of SIRT6. So we were encouraged to look for SIRT6 activators. And today already Professor Zhu and Wang talked about SIRT6 and SIRT6 activators, so many people are looking for that. So what was different about our work, because again, encouraged by our study in centenarians that ribosylation activity was important, we wanted to focus on the activators that activate mono-ATP ribosylation, not the acetylation activity. And we screened several published activators, MDL800 among them, and most of them didn't activate the ribosylation, but there was one molecule, which is a natural product found in abundance in brown seaweed, and this is called Fucoidan, that very strongly activated both activities of SIRT6, deacetylation and ribosylation. So we decided, okay, why don't we test it in mice? And I should say, well, brown seaweed is also a very popular food item, and it's consumed in Asia, in China, Korea, Japan, so it's very safe. At least this we know. We do no harm with it. Another important point here is that Fucoidans, it's not a uniform molecule. There are different versions. It can be branched, it can be sulfated, so not every seaweed produces the same exact molecule. So we had to actually find those isolates that were activating SIRT6. And then we took this version of Fucoidan and administered it to older mice. So we started at 14 months of age. The mice receiving supplement, they were looking nicer, so we could see better coat, less gray hair. And then when we looked at frailty scores in these mice, there was significantly reduced frailty. So that was really exciting. And that continued, of course, as mice were getting older, frailty scores increased, but the difference remained. So we were also interested, like, what exactly are we changing here? And we looked at inflammatory cytokines and also transcriptomes of these mice. So here we are looking at plasma cytokines, and you can see that mice receiving Fucoidan, there was significantly reduced level of inflammatory cytokines. So these mice were less inflamed. Of course, the question is whether Fucoidan is working through SIRT6, right? So it is a very strong activator in vitro, but it's a natural molecule product that may be doing multiple things in vivo. So one experiment trying to answer this question, we looked at SIRT6 knockout mice, and when we gave Fucoidan to them, so these mice, as you already heard earlier today, they're very short-lived. There was really no change by administering Fucoidan. Another thing, I don't have a slide for it because this is actually a very fresh result. We found that these mice, also they had lower levels of expression of transposable elements, which is another function of SIRT6, which again provides additional line of evidence that the effect is mediated by SIRT6. And finally, lifespan. So we actually achieved extension in median lifespan of the mice receiving Fucoidan. So this is, very exciting, and especially to me, I like the fact that how safe it is. So we can recommend to everyone use it. So now, let's talk about whales. Switch gears and talk about something really big. So the whale is really interesting. It's not only the longest-lived mammal, but it's also one of the largest. Bowhead whale is Arctic whale. They can reach 60 tons, even up to 100 tons. So you can imagine compared to human, even compared to elephant, it's really big. So which makes them potentially very prone to cancer because they have so many cells. So there is this concept of Paetos paradox, where statistically you would expect long-lived and large animals to develop cancer more frequently, but they don't because they have some kind of mechanism not to do this. So yeah, so these are my students and postdocs in Alaska when they went to collect bowhead whale cells with the help of local Inuit tribes. So we were interested in addressing Paetos paradox in the whale. So it was addressed in elephants before, and it was found that elephants have multiple copies of p53 genes, some of them pseudogenes, some of them maybe active, but overall in the elephant they were just having enhanced level of apoptosis, and that may be the way how they were fighting cancer. But in the whale, we didn't see that. Actually, if anything, their p53 activity was lower than in human. And then when we looked at the number of oncogenic hits that are required for malignant transformation of whale cell, and we expected that we would need more than for human, again from this idea of Paetos paradox, but we actually needed less. So in human fibroblasts, we require 5 hits from Bob Weinberg's studies, but in the whale, only 4. So the question, okay, how they do it, what's going on? And then we realized that maybe these type of hits, like p53 or RB, they eliminate, these tumor suppressors eliminate cells that already mutated, that already accumulated oncogenic mutations. And maybe if in the whale they just don't get things deteriorate to that point, maybe they don't need to enhance p53, maybe they take care of the genome instead. And indeed, what we found was that in the whale, the double-strand break repair, which again, the same double-strand break repair we found to be important before from comparative study, was much more efficient than in human even. So like, it's really difficult to beat human to anything, but whale does it. And it was also more accurate. So here we are looking at mutations generated at DNA repair site, and whale was much more accurate than human. So when we looked into the mechanism of this, we've identified, again, for the sake of time, I will not go to a lot of details here, but this paper currently is in revision, so I hope it will be accepted soon. So what we found that in the whale, this protein called, here, cold-induced RNA binding protein was present in very large quantities, and first we performed mass spectrometry, of course, and then we just verified by Western blot. So they have maybe 100-fold excess of KIR. which is a protein mostly known for its function in protein translation, where it binds to stress-responsive transcripts in the cytoplasm and promotes their translation. But it's primarily localized in the nucleus, so it must be doing something important in the nucleus. There was one study that showed that it is important at double-strand breaks. So we were quite inspired by that and thought, okay, what if we now take whale KIRP and express it in human cells? Is it going to improve DNA repair? And this is what we've done. So we are expressing bowhead whale KIRP in human fibroblasts. It's expressed very well, and it improved non-homologous end joining pathway of repair almost twofold, and also homologous recombination. So by expressing a single whale protein, we were able to enhance human DNA repair. So this is, I guess, another example of what we can learn from a long-lived species. So to summarize this study, we found that in the whale, they evolved this unique adaptation of upregulating KIRP, which was probably driven by their lifestyle because they live in Arctic and cold water, but then it was co-opted for genome maintenance. And as a result, they upregulate genome maintenance instead of upregulating these tumor suppressive mechanisms that are gatekeepers like P53 or RB, which may be a smart way of doing it, because if you plan to live for 200 years, you don't want to kill too many cells because you may run out of stem cells eventually, but maybe it's better to just maintain your genome. And now what I want to show that sort of a different approach, because here I showed you some examples of where we took one long-lived species and we analyzed mechanisms that just evolved in that species. But what if we look at multiple species in an unbiased way and see what we come up with? So this is the first study we performed was using transcriptomics, and this is the cover where we, if you've seen painting of the Fountain of Youth, so this is our version of the Fountain of Youth. So what we found with the animals was, kind of really exciting. So here, the transcriptomic study, we took 26 species and we analyzed multiple tissues, of which one multiple individuals, and then I will also show you a couple of slides about our ongoing work with the proteomics on an even larger data set of 43 species. So from the transcriptome, again, this is completely unbiased, we just see what pathways correlate with positively with maximum lifespan, and we came up with DNA repair and especially double-strand break repair. So that was really nice. And also we looked at what pathways negatively correlate to maximum lifespan. And here we had a bunch of metabolic pathways, and then there was this really interesting cluster which was related to inflammation, which was mentioned multiple times today as a sort of a hallmark of aging. But here, when we look not at aging, but at longevity, we see that long-lived species downregulate inflammation. And in particular, there is this cluster, cytosolic DNA sensing, which includes genes such as SIGA, STING. They are downregulated in long-lived species. So very interestingly, in bats, some of these genes are just eliminated completely, but in the data set that we used here, we didn't include bats. But still, we have downregulated cytosolic DNA sensing. So you may be wondering, what exactly is it sensing, right, that is important for lifespan? And I will just show one slide where, what is it sensing? And we think it's sensing transposable elements. So we have a really nice story where we showed that if we knock down line 1 transposable elements, we can extend lifespan in mice. And tomorrow, my colleague Andrei Silvanov will tell you much more about it. But now let me just move on to proteomics. So we also perform proteomics on this expanded data set. And some of the interesting finds there, again, I should mention, proteomics is not as sensitive to transcriptomics, so we don't get some of the same like transcription factors. But we get something quite interesting. A very strong cluster was related to extracellular matrix. So already on the example of naked morad, you saw that extracellular matrix is important with hyaluronic acid, but now at a much wider array of species, we also see the same. So let me summarize, so I don't run out of time, what lessons have we learned from long-lived species? Well, first, extracellular matrix is really important. Long-lived species upregulate pathways related to matrix, and especially with naked MORAD, high molecular weight hyaluronic acid, may extend lifespan even for other species. And then, double-strand break repair is really important. It can be achieved by more active SIRT6, or in the case of bowhead whale, we find a different strategy to achieve that by upregulating KIRP, so more stable epigenome is important because SIRT6 also takes care of epigenome and transposable elements, and less cytoplasmic DNA cells. So this is my group. I want to acknowledge people. All of this work was done in collaboration with Andrei Siluanov, and we're always looking for young, talented people who join us, and also would like to acknowledge our external collaborators and our funding sources. And happy to take questions.